Ivy 153. Welcome to today's episode of Plant Stars, where I introduce you to my house plants because we're learning about plants in class. First guest I want to introduce you to is one of my favorites. Actually, they're all my favorite. This is a purple possum passion vine right here. This is a fruit-bearing passion vine. It hasn't produced any fruit yet, but passion fruit are one of my favorites, which is why I got it. And it was a tiny little baby when I got it, and you can see now it's grown a lot. So this is a passion flower vine right here that produces fruit, which will be relevant for today's lecture. The other guest we have is this Norfolk Island pine. It's actually not a true pine. It does have needles like a pine, but it's not a true pine. It is a conifer though, which we will talk about today in lecture 2. Okay, let's get into a discussion of plant structure and function. So first, let's talk a little bit about the two types of seed-bearing plants that you can have. The first one we're going to talk about are gymnosperms. They're called naked seeds. These plants are plants that produce seeds that are not covered by any fruit, and they don't produce flowers. So this includes conifers, pines, fir, and hemlock. So that Norfolk Island pine that I just showed you, that's not a true pine but is a conifer, would be considered a gymnosperm. You can kind of think of the cones that they produce as like it's not a fruit, it's because angiosperms produce fruit, but the seeds are covered in the cone for the gymnosperms. So these are the naked seed plants. We also have angiosperms, which are covered seed plants. These are flower and fruit-bearing plants. The seeds that angiosperms produce are going to be the seeds that become the next generation of plants. Usually you have a flower that then produces a fruit, like a strawberry, or an apple flower that then produces this kind of fruit. This is a great diagram here to show the difference between the seeds that are produced by gymnosperms and angiosperms. And the fruits are going to cover and protect those seeds. The tissue that's in the fruit usually has a high sugar content, which makes them very good to eat, and they are designed that way because animals eating them and then dispersing them in their excrement is really important mode of dispersal for angiosperms. So fruit is designed to be tasty. Most of the information we're going to talk about with respect to plants will involve seed-bearing plants, these gymnosperms and these angiosperms, and you should know the difference between these two types of seed-bearing plants. Let's talk a little bit about the structure of plants as a whole. Plants are divided between what's above ground, which is the shoot, and what's below ground, which is the root. So you got shoots and roots. These two basic divisions will show similarity in the way they grow, but they are significantly different in the activities within each part. So in the shoot portion of the plant above ground, this consists of the shoots and the leaves of the plant. This is the primary photosynthetic center of the plant and is generally the location of the reproductive tissues of the plant, which is the flowers and then the seeds that they produce. All the plants will have one central stem containing the vascular systems, and this is the main support portion of the plant. So that's what you see here. The repeating or iterative units of the vegetative stem will contain internodes that you see here, the stem between the nodes, that's what the internode is, and then the nodes, which are on either end of the internode, and then those are at different locations on the branches along the stems, and then out of each node is where you'll get the growth of leaves on the plant. There are 3 parts to a leaf. There's the petiole, which you see here. That's the base of it, the thin stem extension that supports the leaf. There are the different leaf parts that contain both the flat portion, which is the blade. The blade is the flat portion. And then there's these support structures, which are the veins. So a blade is supported by the veins. And then you have the auxiliary buds, which you see here. This is a dormant or not growing portion of the node that allows for regrowth of the branch or leaf if it's eaten or knocked off the plant. So in some of my house plants, I'd really like them to branch out, and so what I've done on some of them is I've trimmed them back to these nodes so that then when they regrow, they'll grow many branches out of that node. All of the vegetative parts of the plant that grow out of the nodes are not the reproductive portion of the plant, but they will form the photosynthetic center of the plant. So they're really important in growth and development because this is where photosynthesis occurs. The shoot apex, which is all the way up here, this is the leading end of the shoot, and it will form the location of the primary growth of the plant. If this apex is lost or chewed off by an animal, then most plants will respond by allowing one of the sides of the shoots to develop as the new apex, which you can encourage branching in your houseplants by trimming them, and then that encourages new growth. The reproductive portion of the plant, and right here we're talking about angiosperms because remember, gymnosperms do not make flowers and fruit. The flower, fruit, and the seed of the plant will grow directly out of the stem of the shoot, although there is some variability in how they're going to do this. So that's all part of the shoot. Now let's talk about what's going on beneath the ground. That's the root. The portion of, and the root is the portion that grows underground. This acts to anchor the plant to the ground, and it's how the plant absorbs water and takes up ions. You're going to see that there are many different modifications to the kinds of root systems that we'll see when we get into a little more detail on roots. But in general, there are two main primary parts to the root. There are two main parts of the root. There's the primary root, which is the thick and often vertical portion of the root. This generally will grow straight down into the soil. And then you can have lateral roots, which you see here. These are side branches of the primary root. They will grow laterally along the sides of the primary root. And then you have the root apex. So you have a shoot apex, and then you also have a root apex. As with the shoot above, the primary site of growth for the root will be the apex down here, which will form the lowest part of the root. So that's just some general structure of plants, vascular plants. Now let's talk about different types of plant tissues. There are three types of cells or tissues which make up both the root and the shoot. You can have dermal tissues. These are the outermost thick layer of the plant, and they form the outer protective covering of the plant. You can also have ground tissues. These are the middle portion of the plants, and they are the most functional part of the plants because this is where photosynthesis occurs. So the ground tissues are where photosynthesis occurs. And this is where they store carbohydrates and where they secrete hormones. And then you have the vascular tissue. This is generally the deepest layers of the plant, and they're responsible for the movement of water and other material in the plant. And you can kind of think about them as being like the ecto, endo, and mesoderm of the animals, these three different layers. It's just in plants. Now let's talk about the basic structure. So we talked about the structure of a whole plant. We talked about the different tissue layers. Now let's go all the way down to the cellular level and talk about the structure of their cells. So all plant cells are eukaryotic, which means that they'll contain the DNA inside the nucleus of the cell, and they'll contain other organelles. But for the most part, the organelles in the plant's cells will have similar structure and function to the animals. However, there are a couple of differences between the plant and animal cells. Plant cells, for instance, they have a cell wall. So they have a thick outer layer of the plant cells, which will be made up of cellulose, a polymer of glucose, which will act to give the cell structure and protect the cell. They also have chloroplasts. As we already talked about chloroplast a bit, these are those triple membrane structures of the plants, which will contain the chlorophyll and will be the site of photosynthesis in the plant. They also have vacuoles. The vacuoles are the largest central organelle of the plant, and these will be the location of waste storage and will be very important in the regulation of water within the cell. As vacuoles fill with water, the plant cells will be forced against the cell wall, and the plant cell will become very rigid. If the vacuole lowers the amount of water, the plant cell will become limp. And we'll talk a little bit in more detail about this later on in this lecture. you don't have to worry too much about knowing all of the cellular components on this slide here for the exam or for a quiz. This just simply shows how the plant cell will put down layers of cellulose around the outside of the cell to make the cell wall thicker. That's this part here. Plants grow in a very different way than animal cells grow. They have these specialized growth cells called meristem cells. One really important thing to keep in mind when we're thinking about plant cells is that the cells do not move. And that's because they have this really thick cell wall. So you got to think about how having this cell wall limits their movement and how this is going to change the way in which they grow. If an animal needs a cell to replace another, they're able to move through the body. However, for plants, there has to be this ability to form different types of cells in the same locations of the body. To achieve this kind of growth without the movement of cells through the plant contains clusters of cells which will be able to differentiate into many different types of cells located within specific regions of the plant. So these cells are called the meristem cells, and like stem cells in animals, these are cells that are able to be changed into different types of cells in the plant. So they're called meristem cells because they resemble the stem cells in animals that can change into different types of cells. And when these cells divide, only one of the two daughter cells will become specialized. I'm going to say that again because that's important for understanding how they work. When meristem cells divide, only one of the two daughter cells will become specialized. The other cell will remain a meristem cell. This is important because it's going to allow for the growth of the plant while retaining some cells which have the ability to become the other types of cells. And these are just different places of meristem. So you can have apical meristem cells. They're at the apex where they're involved in primary growth, and then you can have lateral meristem cells that are not at the top of the plant that aid in secondary growth of the plant. Oh, so a little more about these apical and lateral stem cells. The apical meristems are located at the apex of the plants, and those are clusters of cells which are at the ends of the plants and are responsible for the primary growth of the plants. So they allow it to grow up in the plant. These are the apical meristem cells. So they're located here at the apex. Each type of plant tissue that will be formed will have its own cluster of meristem cells. You can have protoderm cells, which will form the dermal tissues. I'll say that again. You can have protoderm cells, which will form the dermal tissues. You can have procambium cells, which will form the vascular tissues, and then you can have ground meristem, which will form the ground tissues. And those are all different types of apical meristem cells. And you can see those labeled here on this graph, or on this picture. Then let's get to the lateral meristem cells. These are located on the sides of the stems, coming out of the sides here. And these are going to allow the growth of the stems and the roots to increase in diameter, so going this way. So apical meristems go this way, lateral meristems go this way. Because the primary growth of the plants will be to extend the apex of the plant, the secondary growth will be growth in the diameter. Like I said, these cells will be used in the formation of the branches and the lateral roots. And as a result, that's why we call these the secondary growth cells. Where you can most easily observe the process of this growth in action is when you look at the rings of the inside of logs cut from trees. Each year, the growth and diameter of the tree will cause another ring to be added to the tree. And that is a really great example. The tree rings that you see in a tree when you cut it down, it's a really great example of this lateral meristem cell growth. Also note that the vascular tissue of the stem is formed by the growth of the two cambium layers, which will be located directly under the bark of the tree. You can have cork cambium, which forms the bark, and then you can have the vascular cambium, which forms the vascular tissues. And if you cut completely around the tree and sever those two layers, the tree will die because it can't repair the vascular tissues on the plant. Now let's talk a little bit about plant dermal tissues. These are going to form the dermal layer of the plant. This is typically one layer thick of cells, which will form the protective epidermal layer outside of the plant. They're really important in protecting the plant from water loss or protecting them from too much water absorption and rot in wet areas. And this layer is going to be covered in a thin fatty layer called the cutin. That's here. And that's in young plants. Some plants will even have a thicker waxy layer, such as succulents and cacti. I showed you one of my favorite succulents last time. This waxy cuticle layer is even more efficient at preventing water loss. And you can tell if a plant has kind of a waxy cuticle because you'll see, because that wax is hydrophobic, you can see water beating up on the outside if it's got that waxy cuticle, like in cactuses or succulents. Now let's talk a little bit about guard cells before we move on to some other plant structures. The dermal layer is also going to contain the specialized cells which will allow for opening and closing of the stomata of the leaves, and these cells are called the guard cells. These are evenly spaced cells in the undersurface of the leaves, and they're going to regulate the gas exchange by the plant tissues. The opening and closing of the stomata will be regulated by the amount of water which is in each cell, and the pressure of the water pushing the cell against the cell wall causes a change in the shape of the cells. So the stoma is the opening right here. Let's talk a little bit about the turgor pressure and how that's involved in these guard cells. Turgor pressure is the name of the pressure that changes the shape of the cell as a result of the changes due to pressure. And the change in the shape of the cell will cause the cells to have an opening between them or to have the cell walls tightly pushed together. And when you increase the turgor pressure inside the cells, this is going to cause the stoma here to be opened. So the amount of water inside the vacuoles in the cells will be determined by the movement of ions in the organic molecules into the vacuole. And we'll talk a little bit about water potential and water movement in a little bit. One other type of plant structure that's really important for plants in protection are called trichomes. These are hairy-like outgrowths on the cell walls and cell membrane, and they're not unique cells. They're not cells in themselves, but they're extensions of dermal cells. So they're like little prickly hairs that extend out of a cell. They're not cells themselves. And these are going to give the surface of the leaves a woolly or a fuzzy appearance. And they serve a couple of different roles in the protection for the plant. So they can shade the surface of the leaf, which cools the surface of the leaf. They could also cover stoma openings, which will help prevent water loss when the stoma are open and water might bead on these trichomes. And some of these are secretory, which means that they act to secrete a toxic substance onto the surface of the leaves, and that will reduce the amount of the plant being eaten. And some of these can be particularly hard and are capable of stabbing different herbivores that might try to eat them. And so these trichomes can be really hard for like pest insects to eat through, and so they aid in protection that way as well. Now let's talk a little bit about root hairs. Root hairs are tubular extensions of the epidermal cells on the surface of the root, and they'll be most prominent on young roots. And they'll be limited in the growing plants to the apical surface of the roots. They function a lot in increasing the surface area of the root, and they assist in the absorption of water and ions on the surface of the root. And they show a really similar mechanism of formation as seen in the trichomes, so they're kind of like these little projections that come out of epidermal cells. Do note, though, that these are in no way related to the lateral roots, though. The lateral roots are multicellular. These are just extensions coming out of a cell. They're not individual cells themselves. They're very different. And their main role is to increase absorption capacity by increasing that surface area that's in contact with water and the soil. Now let's talk a little bit about ground tissues. The ground tissues are the cells which will perform most of the functions of the plants, and they'll be the support structures, storage, secretion, photosynthesis, and protection. There are 3 types of these cells. There are parenchymal cells. The most common type of plant cells are parenchymal cells, and they can be characterized as having thin cell walls, large central vacuoles, and they have living protoplasts inside. That is, the cells inside the cell walls are alive, and that maybe seem a little weird to say, but we'll see that not all plant cells actually contain living protoplasts. So I'll go through that again one more time. Parenchymal cells are most common. They have thin cell walls, they have large central vacuoles, and they have living protoplasts inside, and we'll see later that not all plant cells actually contain living protoplasts. Their main function of parenchymal cells is to store carbohydrates and other materials, so they have large vacuoles that aid in that. This is where photosynthesis is going to occur, so you see chloroplasts in here, and then they're also involved in secretion. So this is where you're going to store hormones for secretion when the plant needs them during development. We can also have the Sclerenchyma cells. These are structural cells of the plant that have thicker cell walls. Most will contain living protoplasts, and the cells will generally be long and thin. And they're the flexible support cells of the plant. When you think about the strings that you can get in celery, the parts that are kind of tough and hard to eat, that's the Sclerenchyma. Now let's talk about the Sclerenchyma. These are thicker and tougher cells. That's where that word scleren comes from. And they'll be more rigid structural cells of the plant. These cells will have thick, tough cell walls, and they often contain lignin, which is a really, it's a very dense, hard to break down carbohydrate. This is just an example of what lignin looks like. It's a polymer, a modified sugar molecule, which will form a very hard polymer where it's in the cell wall, and this single molecule by itself may not seem like a lot, but when you put them all together, it can make a really, really tough structure. This is what wood is made out of. Wood is made out of all of this really dense, structurally sound lignin. Unlike other types of cells, the Sclerenchyma cells do not contain living protoplasts when they're mature. So basically, you can think of this as the middle of the wood of a tree is not living tissue, but the cell walls of the Sclerenchyma cells. And so you can have 2 types. You can have fibers, which are linen, these sclerids, the stone cells, and then if you've ever eaten a pear and it has that grit in it, it's made of the Sclerenchyma. So let's talk a little more about the different types of ground tissue. There are 2 types. You can have the fibers, which I just mentioned. These are long and fibrous. You can find this in examples like flax, which can be made into fabric. You can also collect these and cure them to make linen. So some of this fibrous stuff is what we use to make our clothing. There's also sclerids. These are called stone cells, and they'll appear to be hard, small pieces inside the plant. So like if you eat a pear and there's these little gritty pieces inside, like I just said, those are the stone cells. So you have these long fibers, and then you can have these sclerids. Here's an example here. Oh, yeah, here, right here. This is a great picture. So you can have these stone sclerids or these fibers. Now let's talk a little bit about vascular tissues. So there are 2 types. There's the, and they're differentiated by what they carry within their fluids. There's xylem, which carries waters and minerals, and then there's phloem, which carries sugars and hormones. I don't know if you are in lab and you watched that bonus video I made about all of the mealybugs that are living on my plants. Phloem is usually going to be the vascular tissue that insects feed on because it carries sugars and hormones. Xylem, though, on the other hand, that we're talking about now, just carries usually water and dissolved minerals. Xylem is these water-carrying tubes that are inside the plant. There will also be the location of mineral movement in the plant, so anything dissolved in water. You should note on these diagrams the location. In dicot plants, these will be arranged toward the center of the stem. In monocots, these will be located in the center of the vascular bindles, and we'll talk a little bit more about this in the next lecture. The movement of water inside these vessels will only be in an upward direction. That's important to differentiate from the phloem. The movement of water in the xylem will only be in an upward direction, and this will result in the movement of water from the roots of the plant up to the leaves and the branches. Two types of vessels, but they both share one common feature, and that is that the cells will make up the vessels are not living cells, but the cell wall remnants of formerly living cells. The cells will be connected together to form these continuous vessels which carry the water up the plant. And so they function a lot like blood vessels in animals and mammals. So these vessels that I just mentioned, these are the larger cell remnants which will be linked together end to end. And you can see their structure here. And then there's these tracheids. These are thinner cells and longer, and you'll notice the cells are not cylindrical, but will have tapered ends. The vessels that I just mentioned appear to move the water more efficiently and are the more important source of water for the tops of trees. But both types of xylem tissue will contain pits on the side, and this will allow the movement of water between the vascular tissues. The outside of these may be supported by some ground tissues, which will provide support and assist in repair if they become damaged, but generally there's only dead cells that are in these vascular tissues. Now let's talk about the phloem. So phloem is that sugar-rich liquid that a lot of plant-feeding insects and pest insects like my mealybugs like to feed on, because they are carrying the sugars and they're carrying the hormones throughout the plant. Unlike xylem, phloem will move in both directions. So xylem only moves up, phloem moves both up and down, and this will be important to allow the hormones and the sugar to reach all the areas of the plant, because that's the energy and they're also involved in development. Compared to the xylem, the phloem is going to have a more peripheral location in the plant rather than central, and this will be most notable when we talk about Eudicot plants. You can have these sieve cells and sieve tube member cells. These are the elongated cells which will make up the tube which run through the plant and will act to carry the materials through the plant. And the name is derived from the plates at the ends of each cell, which will join the cells together. They have several holes in them, which means it'll resemble kind of like a sieve. So that's these sieve shapes here. And these plates are important in the formation of concentration gradients that are necessary for fluid movement in the tubes. The cells in the phloem are living cells, however, compared to the xylem, and when they're mature, they lack nuclei, which means they can't divide anymore. So they do have living cells inside these cell walls, but they don't have nuclei, so they can't divide anymore. Outside of the sieve tube member cells, there are ground tissue cells. These are called companion cells, which will function to support and assist in the repair of any cells that get damaged in the phloem, like per se by a phloem-feeding insect. Now let's talk about monocots and eucots. These are two different types of vascular plants, and their name is derived by the number of embryonic leaves which form as the
plant sprouts. So monocots have one cotyledon, which you've already learned about in class. That's that early developmental tissue, that embryonic leaf. Monocots only have one cotyledon, whereas dicots or eudicots will have two. That's where these names come from. Dicot means two cotyledons. Monocot means one cotyledon. And although there are other differences between these two types of plants, the one I would like to highlight now is the organization of the vascular tissues. So eudicots, the vascular tissue is arranged in a regular circular pattern around the center of the plant. You can think of the tree again, and the arrangement of the vascular tissues is one of the reasons there's this ability to have that second growth. Monocots will contain vascular bundles, but they're randomly replaced within the ground tissue of the plant. There's no regular pattern, which means the secondary growth of the stems is not able to grow or grow regularly, which limits the ability to grow thick stems. Monocots are really commonly, grasses are typically monocots. Now let's talk a little bit about how all that water moves through this vascular tissue that we just talked about. As I already said before, the xylem is the primary location of the movement of water in the plants, and this will be the movement of water from the roots up the plant into the branches and leaves. Remember, the xylem only moves up the plant. So how does this occur? How do we get water to go from the ground up to the tree, to the top of a tree? The movement into the plant, the movement of water into the plant will occur by two different means. A small amount of water will move into the roots by the action of pushing toward the higher solute concentration inside the root. This will only really be an important method for the movement of water into the roots and not for the movement of water up into the stem of the plant. So that's that pushing movement. And then you can also have pulling, which is transpiration, and that's the movement of pulling water up the stem of the plant. This works through the action of capillary action in the vessels, through capillary action in the stem, and then through the action of cohesion, which is water molecules sticking together because of hydrogen bonding, which hopefully you learned already in Gym Chem, and through adhesion, which is the attachment of water molecules to the sides of the tubes in the xylem that it's moving up. And this will form a stack of water inside the tubes. This enables the water to start moving up the plant. So if you think about how water moved up a straw, if you drink in it, it's very similar. In this case, the water molecules in the bottom of the plant will form a complete line of molecule all the way out the leaf of the plant. When the leaf opens, the stoma, a water molecule can evaporate off the end of the line of molecules, and the movement of a water molecule to replace this will cause the rest of the line to pull water up one molecule at a time. So that's how you get the movement of water, is through the release through these stoma that you can see here. Because this requires the movement of the molecules out the end of the leaf, the water can only move in one direction, which is up. Now let's talk a little bit about water potential. The movement of water will be determined by the water potential that's outside of the leaves. So what is water potential? Water potential is the prediction on the movement of the water in a system based on the physical pressure and the concentration difference. So I'll repeat that again so you can take a note on it. Water potential is the prediction on the movement of water in a system based on the physical pressure and the concentration difference. And as we've already seen with concentration difference, water will only move from a higher potential to a lower potential if you've talked about diffusion before. And if you consider the pressure of water inside the cell, this will help you see how the water will move by transpiration. If you have water at higher pressure inside the cell, the water will push against the wall of the cell to try to move out of the cell. You can kind of think about this as a balloon that's full of air. If you open up that balloon, it'll all rush out the bottom because it's trying to escape from that pressure. That same type of movement will be the physical pressure that's involved in water potential. Solute concentration is the other important factor, so it's a combination of physical pressure in solute potential. Water will move from a lower solute concentration to a higher solute concentration. And in the example shown here, the concentration of, there it is. There's the sucrose. The concentration of sucrose is higher inside the cell than it is outside the cell, which means the solute potential inside the cell is more negative than outside the cell. Since water will move from a higher to a lower potential, the water outside the cell will have a higher potential to move out of the cells. The sum of the physical and the solute potential in the cell will determine whether the plant cell will be filling with water or losing water. So, how does the plant cell actually use this water potential equation? If plant cells fill with water, then the cell will fill the central vacuole with ions. This is to make sure the solute potential inside the cell is more negative, and then the cell will begin to fill with water. This will continue until the physical pressure against the cell walls equals the solute difference, and then water will stop moving into the cell. At this point, the cell will then be full of water, and the pressure will make the cell rigid. So you see this when you have stiff plant stems and leaves are stiff. So terrestrial plants use this water pressure to stand up. If they're floating around in the water, they don't have to do this as much, but they need that rigidity so that they can reach themselves higher up to the sun so they can increase photosynthesis. And so that's why they need to use this water pressure. And they can use those vacuoles to change how rigid or limp their cells are. So what happens if you don't water a terrestrial plant for a long time? Well, then the pressure of the outside of the cell will begin to fall. And even with the higher concentration inside the cell, the water will begin to move outside the cell, and that's when the plant becomes limp. So in times of drought, or maybe you forget to water your houseplants for a little bit, you can see that they kind of limp over because they're losing that internal water pressure inside their cells. Now let's talk a little bit about water potential and cell size. When the cell is full of water, this is where they have high turgor pressure inside the cell. The loss of the pressure inside the cell will cause the cell membrane to pull away from the sides of the wall. This is called plasmolysis. This is loss of water inside the cell and results in plants wilting, as you can see in this poor little plant up here that hasn't gotten enough water. And then turgor, on the other hand, is the membrane against the cell wall, and that's that high water potential inside the cell that helps it look bright and vibrant when you finally remember to water your houseplant. But it's not just water potential and diffusion that acts in this. Biologists noticed that plants were capable of moving water in and out of their cells a little faster than if it were just pure diffusion alone, and that's when they discovered these things called aquaporins. So one way that the movement of water in solutes is facilitated in plants is with these membrane channels called aquaporins. Plant aquaporins are localized in the plasma membrane, as you can see here. They're also in the endoplasmic reticulum, they're in vacuoles, they're in plastids, and in some species in the membrane components interacting with symbiotes that might be growing inside the plant. They're of particular relevance for plants for transporting dissolved gases such as carbon dioxide and ammonia, or metalloids such as boron or silicon. So they're really important for the movement of solutes and for the movement of water, and they allow that movement of water to either create turgor or movement out to create plasmolysis. They facilitate that and make that happen a little faster. That's what these aquaporins do. So how does all of this physics that we talked about with water and chemistry and diffusion and osmosis that we talked about, how does this relate to the movement of water up the plant? So if you consider the difference in water potential in different regions of the plant, in the ground, there will be lower concentrations of solutes in the groundwater, even in the soil, than there is inside the plant, and that means the water potential is much lower inside the plant, and the water will readily move into the roots from the plant. So you always got lower water potential inside the plant, which allows it to constantly be drinking up that water through the roots. Because of the higher ion concentration inside the plants, there's almost always a constant supply of fresh water in the soil. Let me reword that. Because there's a high ion concentration inside the cells, and because there's also a constant supply of fresh water in the soil, the water potential inside the plant roots is much lower, and that's going to allow for that constant movement up. And so that's why in the xylem, you've always got the water moving up, because the water potential is lower inside the plants than it is in the soil. That draws that water up into the roots. They then move up via capillary action and the water potential that we talked about before. And then when they get to the stoma that are in the leaves, that water leaves through that, and it's constantly pulling this water up and out of the plant. Tune in next time to talk a little bit more about phloem transport. That's it for this video. Stay tuned.